Today is Tuesday, November 16th. We are on to our last bit of information on the urinary system that we have to finish up. That should not take the whole lecture time, so we should have time for a question and answer review at the end of this, so it could definitely be useful, especially since it feels like with the holiday, it's been such a long time since we talked about the respiratory system. It was way back in October. And then all leading up till Thursday's exam. Unfortunately, the quiz is already closed, so it's not possible to complete the quiz anymore. But talk to me during the break and we can take care of that. Thursday is your fourth lab and lecture exam. So again, you know what to expect from that. Then you have a nice 5-day break before our next class. So Tuesday, the 23rd. We do have class. I know it is a bit challenging for some of you, especially those of you with kids. I know my kids have the whole week off of school for Thanksgiving. We do not. We only have Thursday and Friday off. So I would love to be able to not give you class on that day, but we only have 4 more lectures, and this is one of them. So we can't miss 25% of the reproductive system. So we will have class. You do have 2 assignments that are going to be due. And then you get to take Thursday off, and then we'll come back. And basically at that point, it's then a quick 3-lecture sprint to the final lab and lecture exam, and then the overall cumulative final exam as well. So that's it. We're getting down to it. Any questions on any of that? All right, excellent. Then let's pick up where we left off in the last class. In the last class, we were talking about the proximal convoluted tubule. And so I've drawn a little bit of it here for you. Oops. Not sure what happened there. Convoluted. There we go. And we were talking about how basically there are 5 processes that take place in the proximal convoluted tubule. The first of which is the reabsorption. And actually, I think for this, I'm going to need to make it smaller. Of organic nutrients. This is things like amino acids, like glucose, like small proteins, things along those lines as well. These, remember, we talked about there are a couple paths that things can take. And in general, this one is going to be transmembrane or transcellular, where it is going to pass through the plasma membrane, and it is going to primarily use active transport. So again, we're going to have things like glucose, things like amino acids, and they are going to cross 2 membranes. They are going to cross the apical surface of our proximal convoluted tubule, the distal surface of the convoluted tubule, into the interstitial fluid, at which, of course, I guess you could argue it has to cross a 3rd membrane, the simple squamous endothelium of the peritubular capillary, to be reabsorbed back into the blood. We are also going to get a reabsorption of our ions. Of course, the most important of these is going to be sodium. As we know, sodium helps us do a whole lot of work. It is the most common of all of those, and then also manganese and all sorts of other things as well. However, we did say for potassium, it is going to be not as important. Because remember, we want to keep low levels of potassium in the interstitial fluid, in the blood plasma. So, and again, this is going to be transcellular. And this is going to, again, primarily use active transport. And again, remember, when I say active transport, we can have primary or secondary, where it can be co-transporters or it could just be pumps. So again, things like sodium, things like magnesium, things like chloride, right? All of these things, et cetera, are going to be traveling back as well. Now, if you remember, as we talked about last time, with all of this stuff going back out of the lumen, out of the filtrate, back into the blood, what always follows stuff?
Water.
Water. And so we talked about how in this case, water has no choice. Water has to follow this stuff that is being drawn back in. And does anybody remember what we called that type of water reabsorption?
Obligatory.
Exactly. Right. Water has no choice in this matter. Our water has to follow all of that stuff. So all of that stuff is going back. Oops, I wanted to make that blue. And so it follows it back as well. And if you remember, we actually mentioned that there is a special type of channel that will be located in the plasma membrane of our cell called an aquaporin. And that aquaporin basically is always present in the proximal convoluted tubule. So water always has to follow all of that stuff back. So we have this obligatory water reabsorption where the water must follow the stuff that is being reabsorbed. All right. Questions on this? Again, I appreciate I'm going a little fast, but I know we talked about this at the end of the last class, so it shouldn't hopefully be too tricky. Now, remember, as water leaves, that ends up as water leaves, and let's actually write this right here. As water leaves, for the stuff that remains, we have the same amount of solutes, but we have less water. So what does that do to the concentration of the stuff that remains?
Concentration increases.
Exactly. The concentration of the remaining materials is going to increase as a result of this. And this produces a concentration gradient. And as we know, solutes like to move down their concentration gradient. And does it require any energy to do that?
No.
No. So what's going to happen is we're going to get actually the passive reabsorption of certain materials because of the increased concentration gradient. Some of this will occur between the cells. Remember, there are those tight junctions that help to limit what can pass between the cells, but they're not able to completely stop things from sneaking their way back in. So sure enough, we are going to have some things that will move paracellularly, sneak between the cracks of the two cells and come back that way. But these cells could also have leak channels as well. And so those leak channels would be another way that substances could get out of the lumen into the cell and then ultimately into the peritubular capillary without having to use energy. So it occurs paracellularly or via leak channels. And so the key with this is, again, no energy is used. And for certain ions, for instance, as much as 50% of those ions may actually be reabsorbed this way, paracellularly or through the leak channels, passively through these. And then, of course, we said there were 5 things. What is the 5th thing that happens here in the proximal convoluted tubule?
Tubular secretion.
Yeah, exactly. We are focusing more on the movement of stuff back into the blood supply. But remember, the proximal convoluted tubule is where most of the secretion occurs as well. Basically, it's just reabsorption in reverse. It is primarily active, meaning that it's going to use ATP. It just moves in a different direction. So this is how small, tiny stuff like urea. Your book does a really excellent job of talking about urea and what happens with urea. Urea is a really interesting ion that, organic molecule that we're not going to have a lot of time to talk about, but it plays a huge role in maintaining the osmolarity of our blood. We think of it as a waste the same way we think of carbon dioxide as a waste. But remember, our goal in our respiratory system wasn't to get rid of as much of the CO2 as possible, but to have the appropriate amount. Same thing with urea. Urea is a metabolic waste, but we don't want to get rid of all of it. It plays a huge role in maintaining the osmolarity of our blood. So it is constantly being pumped out in places like this, but it's also leaking back in as well. So it's constantly being circulated throughout the nephron to really have just the right amount of it inside of our blood. So those are those 4 pathways. We've done it here with the pretty picture. And like I said, we did this and talked about this in the last class as well. So again, organic nutrients, our ions, bringing those things back, our reabsorption via those aquaporins of that obligatory water reabsorption, which increases the concentration for our passive reabsorption through those paracellular and leak channels, and then also secretion.
Could you go back to that image, please?
I'm sorry?
Could you go back to that image, please?
The one I drew?
Yeah.
Yeah. All right, so these are the things. Now, just like we saw in the digestive system, there are some examples of these that we want to talk about. All right, so if we are comfortable with these 5 general processes, let's talk about some specific examples. So again, now that we're back at this picture, let's talk about some of that. So any questions on this? Because I need to erase some stuff and modify this stuff. Anything else on this before we move forward? All right. All right, so then let's do this. I need to get rid of all of my arrows. Nope, didn't want to get rid of that. There we go. Perfect. All righty. Not surprisingly, one of the most important substances that is reabsorbed is sodium. As we know, sodium is a major workhorse. It produces our neural action potentials, our muscle action potentials. Its huge driving force allows us to do a tremendous amount of work. So sodium is vitally important for this process. So not surprisingly, sodium is going to be actively moved. Oh, so again, let's think about what's happening on the apical and the basal surface. So when it comes to the apical surface for sodium, do we want to just hope that it comes back in?
No.
No, of course not. So one of the major things, nope, I don't want to use orange. One of the major things we're going to do is we are going to have sodium pumps. Sodium pumps, of course, directly use ATP to bring sodium into the cell. Once we take the sodium out of the lumen and into the cell, it is now saved. So do we just let it accumulate inside of the cell and let it passively leave the distal or basal surface? We could, if you think about it, that's what we did with things in digestion. But remember, sodium allows cells to do work. And if a cell has a tremendous amount of sodium inside of it, is it necessarily going to be easy for it to do work?
No.
No. So not only are we going to use ATP on the apical surface, but we're also going to use ATP on the basal surface. Primarily with an old friend of ours, the sodium potassium ATPase. Sodium potassium ATPase is a pump, which means it directly uses, it directly uses ATP. And it uses that ATP to do what?
Kick 3 sodium out and bring 2 potassium in.
Perfect. It is going to kick 3 sodium out of the cell, and it is going to bring 2 potassium back in. Notice because of this, even though we're bringing in a massive amount of sodium, because of this, we end up with an incredibly low concentration of sodium inside the cell. Now, doesn't that also mean we're going to have an incredibly high concentration of potassium inside the cell? If I'm moving out a massive amount of sodium, doesn't that mean I have to be moving in a massive amount of potassium?
Are there pumps to kick out the potassium?
We don't even need pumps, but you absolutely have the right idea. What we have instead are leak channels. When that potassium comes back in, when it is pumped into the cell, it can very easily just leak back out to be pumped back in again. So notice we're basically able to cycle the amount of potassium, not majorly change the amount of potassium, but we are able to constantly pump sodium out, giving us that huge concentration gradient for sodium. And why might we want to have such an incredibly low concentration of sodium inside of our cell?
To do work with a high concentration gradient?
Exactly. It's going to allow the cell to do work. Remember, one of the works we want to do is we want to reabsorb of our organic, oops, so I spelled it right, of our organic nutrients. So we never finished this. Let's go back up here and write this out. Primary active transport. Primary active transport. We can use that sodium to do work for us. So we are going to, on the apical surface, we are going to primarily use cotransporters. And in particular, we are going to want to use symporters. What do symporters do?
Don't they move 2 molecules at a time in the same
And
direction?
2 molecules in the same direction, exactly. So in this case, notice we have a massively low amount of sodium on the inside. So sodium really wants to come in. So we're all right, sodium, come on in. You really want to come inside? Come on inside. But when you come on inside, you have to bring that glucose, or you have to bring that amino acid with you. Notice back up here to sodium reabsorption, that means that we also use secondary active transport to bring sodium into the cell. Of course, that does bring more sodium into the cell. So what do we have to do about that?
Kick it out.
And don't we already have something in place that allows us to do that? Yep. So we're able to kick out that sodium so that it helps us that way. And then we have our organic nutrients. Here inside the cell. Once it's here inside the cell, it's not in the lumen and we're not going to lose it anymore. So do we need to bother using energy to get it out the passive, get it out the basal surface?
No.
No. So we can pretty much just use facilitated diffusion. To get it out the basal surface. Oops, that should have been a 2. Excellent. Notice I've done an amazing job of drawing this here, but we have the pretty pictures from your homework. I mean, from your textbook, shows the same things. Notice here our sodium is actively being moved across the apical surface, but also actively being moved out of the basal surface. Of course, as we talked about, that does give us an increase in potassium inside the cell, but we have leak channels that allow it to leave. And the advantage of this system is twofold. Obviously, we get back all the sodium, or a lot of the sodium that we need for the blood, for the interstitial fluid, but a huge advantage is that it keeps that concentration of sodium inside the cell very low. And as we know, cells love to use sodium to do work. So here is our sodium transport, how we get sodium back into the interstitial fluid, which of course then gets it into the blood. And then, as we said, we can then, whether it's glucose, whether it's amino acids or other organic solutes, as we see here, once again, we see these 2 pathways. Let's go back to the first one again. Sodium is being pumped inside, with a co-transporter, with a primary active transporter being brought inside. Of course, as soon as it gets inside, we have to pump it back out. When it brings in that glucose, once that glucose is saved, then again, we don't have to worry about using ATP to get it out the basal surface. So at the basal surface, for our glucose, for our amino acids, for all those things, we will use just facilitated diffusion. All right. Questions on that? This doesn't make sense. It's about to get worse. So make sure you tell me, does this make sense? Questions on these pathways. They're not too dissimilar to what we did in digestion, so hopefully it makes sense, but I need to know now because it's about to get worse. All right, let's go and look at the third possible pathway that could be an essay question on your exam. Remember, one of the major functions we talked about for our urinary system is helping to regulate the pH of the blood. And there is primarily two ways we do this. One of the ways we do this is with the reabsorption of our bicarbonate ions. Oops. And with the secretion of hydrogen ions. So let's start with the easy one first. I have hydrogen ions here inside. Oh, I may have to make this smaller. I have hydrogen ions here inside of my cell that I need to get rid of. What would be a convenient way to get rid of hydrogen ions here inside my cell?
Antiporters.
Exactly. Antiporters do what?
Kick one molecule in or out and bring one in.
Exactly. So once again, we are going to let a molecule come inside the cell. And when we let that molecule come inside the cell, it's going to do work for us. And the work is going to be kicking out a hydrogen ion. And what would be a good molecule that we could use to allow in to kick out hydrogen?
Sodium.
There we go. So mission accomplished. We've kicked hydrogen ions out of the cell. Mission accomplished. However, you've got to remember that out here in our lumen, we have filtrate. And filtrate contains, among other things, water and dissolved gases like carbon dioxide. Now, we know that carbon dioxide, some of it stays dissolved in the water, but what usually happens with carbon dioxide when it comes in contact with water? What does it most of it do?
Converts to carbonic acid.
Exactly, carbonic acid, H2CO3. It becomes carbonic acid. Of course, carbonic acid, being an acid, doesn't stay in that carbonic acid form. What does it do?
That then converts to hydrogen ions and bicarbonate.
Hydrogen ions. And bicarbonate ions. Excellent. All of that sound vaguely familiar? And what do we know about this type of chemical reaction?
It's reversible.
Yeah, this one is reversible. It can go in either direction. But notice, I just pumped a bunch of hydrogen ions into my lumen. So my concentration of hydrogen ions is going to increase. And when that occurs, does that affect this reversible equation?
Of course.
Yeah, which way does it drive the equation then?
In the reverse direction.
All right. So that increase in hydrogen ion drives the equation to the left in this case. And when we drive the equation to the left in this case, we get more water and we get more carbon dioxide. So we get an increase in concentration, or really the partial pressure. Increases here in the lumen. Now, gases like to be at equilibrium, just like solutes like to be at equilibrium. So if there is a higher amount of carbon dioxide gas in the filtrate, is that carbon dioxide going to want to stay there in the filtrate?
No.
No. Carbon dioxide is a small non-polar molecule. So what does that mean?
It passes through the lumen through simple diffusion.
Excellent. Carbon dioxide enters the cell via just simple diffusion. So now notice we have an increase in carbon dioxide. Again, that in and of itself wouldn't be a big deal, except what else do we have inside the cell? How about water? And when that increased carbon dioxide comes in contact with the water, what does it do? It should be getting easier. There you go. I like that, Amy. Absolutely. We have to redraw our chemical reaction and it's going to draw it to the right. Absolutely. It's going to become carbonic acid, which is going to disassociate into hydrogen ions and bicarbonate ions. And now this time it's going to, and I'll change colors just to emphasize this. Because the carbon dioxide increases, it drives the equation to the right. And as it drives the reaction to the right, we get more hydrogen ions and we get more bicarbonate ions. What are we going to do with those hydrogen ions?
Kick them out.
Get them out. And what do we want to do with the bicarbonate ions?
Bring them in.
Bring them in. Notice, I appreciate my drawing may or may not be easier to see than this, but notice here we see that same type of process. Notice it starts with our hydrogen ions being kicked out. Hydrogen ions leave the blood, enter into the cell, and can be kicked out via that antiporter. Oops. Once in the filtrate, it mixes with the bicarbonate to form carbon dioxide. So again, this drives the increase in hydrogen ions, drives the, let's just get rid of that first arrow, drives the equation in this direction. That allows carbon dioxide to be produced, which can just enter via simple diffusion. Where it mixes with water, forms bicarbonate and hydrogen ions. Those hydrogen ions can then be kicked back out, and our bicarbonate needs to be taken out. Now notice, for our bicarbonate ion, do we want bicarbonate just hanging out inside of the cell?
No, we want it in the blood.
Right. Not only do we want it in the blood, but if you think about it, if it stays in the cell, as bicarbonate levels increased, it might drive the chemical equation back in the other direction. We don't want that either. So notice we are going to, and I know this looks a little tricky down here, but this is actually a co-transporter where we are getting the, probably not a co-transporter, a pump. This is actually a pump that is directly using ATP to kick both the bicarbonate and the sodium out of the cell. So yet another way we get sodium out of the cell to keep those sodium levels low, but we also are able to get that bicarbonate back into the blood as well. So I know it looks like a co-transporter in your book. That illustration I just showed you labels it that, but this is actually a pump where it directly uses ATP to move both sodium and bicarbonate out of the cell, because those are two things we don't want to stay in the cell. So it's okay to use ATP to get them out. And there you go. Just that simply, we've looked at 3 of the very common pathways that things are going to be reabsorbed in the proximal convoluted tubule, which always makes it likely that one of those 3 will be an essay question. All right. Questions on that?
Yeah, I had a question. I think I missed it, but how does the hydrogen ions exit again?
They exit the cell via a antiporter. So this is an antiporter. Antiporters are secondary active transporters. They do not directly use ATP. Instead, they use the energy of another molecule to do work. So in this case, they are using the energy of sodium wanting to come in to kick the hydrogen out. However, remember, these antiporters only continue to work if we later use energy to get rid of that sodium, so sodium levels stay low. So both, this is a symporter, and this is an antiporter. Both of them are secondary active transporters. They do not directly use ATP. They're using the energy of sodium, but we do have to use energy to get rid of sodium so that these will work. Does that make sense?
Yeah. So the symporter, does that use amino acids as well?
It's not using amino acids. So the way I describe it in 430 is if I'm standing, right, things like to move down their concentration gradients. So for instance, if I'm at the top of the hill and you're at the bottom of the hill and I have a ball, I don't have to use any of that energy to get the ball to you. I can just let the ball go. And because it is at a higher potential energy and it wants to be at a lower potential energy, it will roll down the hill to you, right? And I don't have to push it, or I don't have to shove it. I don't have to use any energy. I just have to let it go. That part makes sense?
Mm-hmm.
Okay. Now, if I wanted to give you a message, I could use my energy to yell it at the top of my lungs. I could use my energy to walk down and hand you the piece of paper, or I could merely tape the piece of paper to the ball. And then when I let the ball go, the ball does the work to bring you the message. And that's exactly what's happening here. In this case, sodium really, really, really wants to come into the cell. And so I say, okay, sodium, I will let you come down the cell. You can roll down the hill, but when you roll down the hill, you have to do work for me. You have to bring in this amino acid, or you have to kick out this hydrogen ion. It's kind of like a water wheel. You know, those mills or granaries would put their granary next to a river. Then they'd put a big wheel in the river. And as the water moved down the river, it would turn the wheel, and I could capture that energy to saw my wood or to grind my flour or whatever it was. I'm using somebody else's energy to do work. And that's what these co-transporters do. They use somebody else's energy to do work. But that water wheel only works as long as the river's coming down. These symporters and antiporters only work if sodium really wants to come in the cell. And if we left the cell fill up with sodium, sodium's not going to want to come in anymore, and we can't do any work. So what do we have to do to keep these co-transporters going? We have to find ways to, we have to find ways to kick sodium out of the cell so that it still wants to come in and it will still do work for us.
Gotcha. I appreciate it. Thank you.
Yeah, no problem. Yeah, these are all concepts. I know it was a long time ago, chapter 3, when we talked at cells and membrane transport. So if any of this is a little rusty, it's good to go back there. But hopefully in a nutshell, that helps to make some sense of it. All right. Any other questions on that? Excellent. And so here's another pretty picture that, again, kind of shows all these different pathways and processes that we talked about. And more tables from your textbook and everything else. All right. Like I said, I know we haven't talked about secretion, but we'll save that for the end. But notice by the time our filtrate reaches the end of the proximal convoluted tubule, it has been changed dramatically. 100% of the glucose has been reabsorbed. 100% of the amino acids have been reabsorbed. 65% of the sodium, 65% of the water, 60% of the chloride, 55% of the potassium. At this point, most, if not all, of the useful things have been reabsorbed. Basically, what we're left with is 1/3 of the volume we had before. And it is primarily at this point just water with ions and wastes. That's pretty much all that is left. Now, that doesn't mean there isn't still work to do, because remember, we started with 200 liters. We're now down to 67. But again, we don't produce 67 liters of urine during the portion of the day. So we still have to bring back this water and some of these other solutes that are important. All right. As I mentioned, we can't forget about tubular secretion. Remember, that is reabsorption in reverse, primarily occurring in the proximal convoluted tubule. This is where we get rid of substances that are bound to the plasma proteins. This is where we get rid of some of those metabolic wastes like urea and uric acid that are small things that keep getting reabsorbed. This is where we can get rid of excess calcium and excess potassium and excess calcium. And this is where we, again, get rid of that hydrogen ions to help to maintain our blood pH. And remember, when we're removing those hydrogen ions, as we've seen, that also leads to the reabsorption of bicarbonate, which, as we talked about, is an important blood buffer. So again, it's really kind of the same mechanisms. It's just the different substances moving in different directions. All right. This brings us to the loop of Henle. Like I said, 60% or more of the filtrate has been reabsorbed in the proximal convoluted tubule. So what we're left with now is dealing with it in the nephron, in the loop of Henle. If you remember, we had this issue before of obligatory water reabsorption. What was that again?
Water following stuff.
Yeah, and not only following, but it must follow this stuff as it's being reabsorbed. So if we think of it this way, if we start with a solution of 1 liter of blood plasma, has about 300 milliosmoles. Of solution, and we bring back 50% of the stuff, 50% of the water is going to follow as well. And what we're going to have after that process is half a liter of filtrate. And what would the osmolarity of that filtrate be?
It will be the same.
Yeah, it's going to be the same. There's no way for us to be able to unlink these two in the proximal convoluted tubules. Wherever stuff goes, water is going to follow. But do we necessarily want our urine to have the same amount of stuff in it that our blood does?
No.
No. So we need a way to decouple this. We need a way to separate the reabsorption of ions and the reabsorption of water. And that is what we're able to do in our loop of Henle, in our nephron loop. We're able to do this using a countercurrent mechanism. Anyone know what I mean by a countercurrent?
Isn't it when you have one substance flowing one way and then like right in like a tube and then like right next to it another tube with another substance flowing another way? I think it's usually done for like getting rid of heat.
Absolutely. That is a beautiful example. Absolutely. What'll happen is, for instance, you see this in engines, you see this in nuclear power plants. You have incredibly hot water that is being warmed up by the nuclear reaction that is taking place inside of that nuclear power plant. However, do you want to just dump that hot water out into the environment? Okay, there should have been less pause in that. Absolutely no, you do not. So what they do is they have a second pipe right next to it of cold water. Hold on. Of cold water that runs right next to it. And so we have this cold water that enters the pipe next to it. This cold water absorbs the energy from the hot water. And so by the time that radioactive water leaves, it is now cooled. And the water, the clean water that was in the pipe, the second pipe, is now hot. And this one can now be dumped out into the lake. Of course, the hot water still kills the fish anyway, but still. They call this a heat sink, where you have these two things move in the opposite directions, and that opposite movement basically drives the reactions that takes place. And that's exactly what's happening here. We're having these two opposite processes occurring, and they're going to drive each other. We have the two sides of the loop of Henle, the descending and the ascending. But remember, the descending was also the thin segment, and the ascending is the thick segment. So in that thin descending segment, that side is only allowed to reabsorb water. Water can be reabsorbed, but solutes cannot. Whereas in the thick ascending limb, our ions are reabsorbed and water cannot follow. So here we have two opposite flows of our filtrate and two different processes that drive each other. Let's see how this works. Let's go to the whiteboard and draw this. All right, give myself plenty of room here. Here's my loop of Henle. Thin segment on one side, thick segment on the other. And again, my thick segment is made up of cuboidal cells. Oh, I lied. I want to put that on the inside. And then, of course, then we go into the distal convoluted tubule, and this came from the proximal convoluted tubule. This side is our thin cells. We'll do it on the inside too. So our simple squamous cells. And so, as we know, over here, we have our thin descending limb. And as we mentioned, this thin descending limb is impermeable to solutes and permeable to water. Whereas over here, we have our thick ascending limb. And as we mentioned, it is permeable to solutes, but it is impermeable to water. This is our distal convoluted tubule up here, and this is our proximal convoluted tubule here. And let's, of course, assume that this is a juxtamedullary nephron. What does that mean?
Has a vasorecta.
Okay, that is one thing that it means. What else does it mean? That is absolutely true. What else?
It continues down the medulla.
Absolutely. So we need to also put a line here to distinguish between our cortex and our loop of Henle of the juxtamedullary nephrons. Remember, go deep into the medulla. So we have that nice border to emphasize that as well. Last thing we need to do, as we mentioned, our filtrate. I guess we can make this green. As it is entering into our proximal convoluted tubule, remember we said that the osmolarity of it is 300 milliosmoles. And based on that, do you have any idea what the osmolarity of the interstitial fluid might be in the cortex? Well, since things tend to seek equilibrium, what do you think it's going to be?
300.
There you go. Perfect. Excellent. So here is our starting point. Questions on this? All right, let me take a look at this real quick. All right. All right, yeah, perfect. That's where we want to be. All right, excellent. So we have our starting point, but looking at the time, I want to come fresh at this information. So let's go ahead and take our first break here because we've been at it for an hour. So let's take our first break. We'll take a 15-minute break, come back at, we'll call it 1:20, and we will restart at that point. All right, any questions? And I'll start the recording at that point. Any questions before we take our first break? All right, I'll see you in 15 minutes. Any questions before we get started? All right, stunned silence. Excellent. That's what I love. All right, so as I mentioned, our goal here is going to be to decouple the movement of water and the movement of solutes. And that is what we're able to do here in the loop of Henle, which allows us to make more modifications to our filtrate, which is what we want to do. We know the filtrate as it enters into the loop of Henle is at 300 milliosmoles. But like I said, on the descending thin side, water can be brought out. And on the thick ascending side, our solutes are brought out. Now, the moving of solutes is, again, an active process, especially on the proximal surface. So here on the proximal surface, we have pumps. And these pumps are actually going to move sodium, chloride, and potassium. Primarily sodium and chloride, but also, wow, that's huge. And some potassium back into the interstitial fluid. Now, as solutes are actively pumped, oops. As I mentioned, the wall of our plasma membrane doesn't have those aquaporins. So here now, again, our cells are impermeable to water. So the net effect of this is that we have the same amount of water in the filtrate, but we have less solutes in the filtrate. So what kind of an impact is that going to have on the osmolarity of our filtrate? If we have the same amount of water, but we keep taking stuff out of it, what happens to its osmolarity?
Goes down.
Yeah, exactly. So let's change the color of this to emphasize this. This decreases the osmolarity of the filtrate. We are no longer stuck by whatever is in the interstitial fluid, right? Because with the osmotic forces, we're always up here, up here. The osmolarity of the filtrate always equals the osmolarity of the interstitial fluid. Again, because of that obligatory water reabsorption, these are always going to be equal to each other. But now that's not the case anymore. And in fact, by the time the filtrate reaches the distal convoluted tubule, it is actually down to about 100 milliosmoles. It is a third of the osmolarity. It has a third less stuff in it that it had when it started in the proximal convoluted tubule. So as our solute moves up, pardon me, as the filtrate its osmolarity decreases. All right. We comfortable with that so far?
Yeah.
All right. Now there is a flip side to this. Notice the flip side to this is, as we mentioned on the ascending limb, solutes are being pumped out into the interstitial fluid. And as they're being pumped out into the interstitial fluid, there's more solutes in the interstitial fluid than there were before. So what happens to the osmolarity of our pyramid? What's happening?
Osmolarity increases.
Yeah. Excellent. And here's the last thing. Do you think that the same amount of solutes are pumped out near here, down near the bottom, than there are near the top?
No.
No. Notice, because there is more solutes down here, there's more solutes to be pumped out down here, and there are less solutes to be pumped out up here. What ends up happening is this actually produces an osmolarity gradient. In the pyramid. At the very start of the pyramid, here, I guess I'll sneak this stuff over here so I have room to draw this. Up here near the top, the osmolarity is going to be 400 milliosmoles. And then down here, it may be 600. And then down here, it may be 800. And then down here, it may be 1,000. And then down here at the deepest part, it can actually get as high as 1,200 milliosmoles at the deepest parts of the pyramid. And actually, you know what? I'm going to cheat. Let's do this on the other side. Move this here. kind of fun. I can't never do this on the whiteboard in class. All right, so get out of my way. There we go. Excellent. So here in our pyramids, we get this osmolarity gradient that is produced because more solutes are being pulled out as it ascends. So it gets less and less and less and less. And that means more and more and more ends up in the pyramids. All right. With me so far? All right, stunned silences mean yes, we totally get this. Let's move on. All right. So now let's look what's happening on the descending limb. We have that water, the filtrate that is entering in here. And remember, as we mentioned, when it first enters, it's at 300 milliosmoles. However, as it is moving down inside of the nephron loop, it becomes surrounded by a piece of tubule that is at 400 milliosmoles. And as we talked about, this portion of the loop is permeable to water. Solutes can't move, but water can. Again, we have those aquaporins that are going to allow water to be able to be moved out. Now, notice at this point, there is more stuff in the interstitial fluid, and the stuff wants to move into the tubule, but is stuff able to move into the tubule?
No.
No, because it's not permeable to that. However, water also likes to go to where there's more stuff. So what is water going to do? Not a trick question. Water wants to go to more stuff. Where's there more stuff?
Water's gonna move towards the interstitial fluid.
Exactly. Water leaves the tubule to go to where there's more stuff. And it's going to keep leaving until what happens?
It reaches equilibrium.
Yeah. So a lot of water leaves. Notice what's happening here. We have less water in the filtrate, but same amount of stuff. So if we have less water in the filtrate and the same amount of solutes in the filtrate, what happens to the osmolarity of our filtrate?
It increases. Does it increase to 400?
Exactly. Remember, it's going to go to reach equilibrium. And so indeed, equilibrium at this point is going to be 400 milliosmoles. So water leaves until it's at 400 milliosmoles. But then guess what? Our water keeps moving down. And now our filtrate is at this location and it's at 400 milliosmoles. But what is it surrounded by?
600 milliosmoles.
So what's going to happen?
Water's going to be drawn out.
And it's going to keep being drawn out until what happens?
Reaches equilibrium.
Exactly. So notice what's going to happen is water continues to leave, water continues to exit the filtrate. Because it wants to reach equilibrium. And every time water leaves, the filtrate gets more concentrated. So as the filtrate continues to go down, then at here, it's going to be 800 milliosmoles. Down here, it's going to reach 1,000 milliosmoles. And here at the bottom of the loop of Henle, our filtrate is at 1,200 milliosmoles. Very, very concentrated. Which means it has a lot of solutes. Which we can very easily pump out. We have all these active pumps. Remember, these active pumps are using ATP. So unlike this side where it is passive, and we are stuck by what the osmolarity of the environment is, are we dependent on the osmolarity of the environment on this side if we're using ATP?
No.
No. So notice as we start pumping things out, then at this point, it may reach 1,000. At this point up here, it may reach 800. At this point here, it may reach 600. At this point here, it may reach 400. At this point here, it may reach 200. And at this point here, as we mentioned, finally, it's going to reach 100 milliosmoles. I know this seems like a lot of work, a lot of excessive work to do, but the key that you have to remember about this is that the advantage of this is basically we are able to reabsorb more solutes and more water without the two affecting each other. Remember, with our obligatory water reabsorption, where one goes, pretty much the other goes because of the force of osmolarity. This cancels out the force of osmosis. We are able to decouple these, bring water back separately, bring our solutes back separately. And the net result, or the final result, is, oops, I'm not going to have space to put this, so I'll have to make it small. And I'll make it yellow because that amuses me. Is our final result is going to be a very small volume of very dilute filtrate. There we go. All right, we did it here on the board. Let's look at the pretty pictures from your textbook. Again, we started our explanation of this in the ascending limb because I think it makes a little bit more sense because we see how we set up that concentration gradient, that osmolarity gradient that drives the water movement. Remember, I have simple cuboidal epithelial cells that move sodium and chloride and potassium. Notice they're pulling sodium, chloride, and potassium out. notice there are leak channels where potassium can then sneak back out. So again, they're actively being pumped in, but as we talked about, potassium movement is not as important as the sodium and the chloride. But they do pump all of those solutes out. And again, unlike what we saw in the proximal convoluted tubule, when we're bringing back all of these solutes, water does not follow. And that has two effects. That basically lowers the osmolarity of the filtrate in the lumen, and it raises the osmolarity in the interstitial fluid. More solutes are pumped out near the bottom of the nephron loop than at the top. So as a result of that, we get this osmolarity gradient. Starting at 300, where it's equal to what it is in the cortex, but it can quadruple, getting as concentrated as 1200 milliosmoles. So again, notice the two big effects of this. It extremely dilutes our filtrate so that we produce dilute urine, and it sets up this concentration gradient. And this osmolarity gradient, this concentration gradient, this osmolarity gradient is vital for the movement of water. And that's what happens on the thin descending limb. And again, there we see the changes in osmolarity. And there's our nephron loop that we were talking about. On the thin limb, simple squamous, permeable to water, but not solutes. So even though solutes want to come in, they cannot. Instead, what happens is that our water leaves. And because this is a passive process, it leaves until it reaches equilibrium with its environment. So as it travels down the loop of Henle, water leaves, solutes amount stay the same. And so our solute, our filtrate, becomes more and more concentrated as it travels down that thin descending limb. And that gives us that massive concentration that allows us to then pump massive amounts of sodium and chloride, and to a lesser extent potassium, out of it on the way out. And this process continues. Questions on this? This doesn't make sense. It's just about to get worse. So let's make sure this makes sense. All right. So the last thing that someone mentioned earlier, so we'll mention it again, is remember that with these juxtamedullary nephrons, they have a vasorecta, which, remember, we mentioned was this long draping capillary system that sits over the top of this. And this capillary system is important because it produces another counter current mechanism. So let's think again about what's going on here with our filtrate. We know our filtrate is, whoops, no, well, it doesn't matter now. our filtrate is going down on this side and it is coming up on this side. As it is going down on this side, we know that water is leaving the filtrate. And also we know that our pyramid becomes more, gets a higher form, let's say it this way. Pyramid forms that osmolarity gradient. So we know, for instance, that, actually, let's do it over here. No, crap, I don't know where to put this. Or, no, sorry, because of the osmolarity gradient. And we know on this side, on the ascending side, solutes leave the filtrate, and that produces the osmolarity gradient. And so, as we mentioned, our gradient is about 400 on the top. Well, let's say 300 and 1200 milliosmoles that way. And our solutes are being moved out. Now, if I have solutes moving out one side and water moving out the other side, wouldn't that combination of water and solutes equalize the osmolarity? Isn't that how things work? Yes, we add a little bit more solutes to it, and that makes it more concentrated. But then when we add more water to it, it would then dilute it. And so the free movement of these solutes and water could disrupt our osmolarity gradient, and this wouldn't work anymore. So the goal of the Vasorecta is to use that countercurrent mechanism to maintain the osmolarity gradient. And the way it's able to do this is that the Vasorecta is equally permeable to water and solutes. So let's take a look at a piece of the Vasorecta. And we'll just do it over here on this side. The filtrate is moving up on the ascending limb. So the portion of the Vasorecta associated with the ascending limb, not surprisingly, the blood moves in the opposite direction. So we have our counter current mechanism. Now, remember, as our blood, like all the blood in our body, starts at 300 milliosmoles, as it's traveling downward, and let's say, for instance, it goes to an environment here in the middle where it's at 600 milliosmoles. At 600 milliosmoles in this location, which way do my solutes want to move? If I'm starting at 300 here inside of the Vasorecta, so I have an osmolarity of 300 inside and 600 in its surrounding environment, which way do solutes want to move? Come on, guys.
Move out.
Things move down their concentration gradients. So if you got 600 of something on one side and 300 something on the other side, which way are things going to move?
Towards the lower concentration.
Yeah. So in that case, is that into the blood vessel or out of the blood vessel?
Into it.
Exactly. Solutes move into the blood vessel. And as the solutes move into the blood vessel, which way does the blood carry it? The blood moving down or the blood moving up?
Down.
Yeah. So notice it basically what our blood vessel is doing here is it picks up solutes and it brings them to the bottom of the loop or bottom of the pyramid. Also, what about water? Which ways water want to move?
Up.
Well, it wants to move out. And again, I'm still thinking of the same
Oh,
place right here. So again, we're talking about at this location right here, right? Water wants to move out of the blood vessel. So notice as it moves down, basically what our Vasorecta does is it gives water to these upper areas and it's picking up the solutes and bringing these solutes down to the bottom. So it releases water as it goes down and it picks up solutes as it goes down. And if we're giving more water at the top and we're bringing more solutes to the bottom, is that going to help to maintain the concentration gradient?
yeah.
Yeah. And notice the exact same thing happens over here. We have that same concentration gradient. We have our blood moving up on this side. And notice as it goes from an environment where it was at, for instance, 1200 to an environment, let's move it up a little higher, to an environment where it was 600 in the interstitial fluid. Then notice here the opposite's going to happen. It is going to give off solutes. Oops, sorry. It is going to, as it goes off, it gives off solutes. And as it gives off solutes, it picks up water. And as it picks up that water, it brings that water up towards the top. So by producing basically these little circuits, essentially that's what it's doing. It's picking up salt and bringing it down, picking up water and bringing it up. And by having these two alternating counter currents, this helps to maintain the osmolarity gradient. And again, if you don't like my drawing, if I've been confusing to you, then again, here we have that pretty picture from your textbook that talks about this countercurrent mechanism of the Vasorecta, what we call the countercurrent exchanger, where again, as it goes down, it is picking up solutes and bringing those solutes down to release them. And then as it goes up, it is picking up water and it releases water on its way down. So because it's permeable to both, this movement of both ions and solutes allow us to maintain that concentration gradient in the pyramid. All right, you guys got all quiet on me here, but questions on this?
No.
So how does how is the gradient set for the different type of nephron?
Great question.
One without a vasorecta. The name's escaping me.
The cortical nephrons.
Thank you.
You are correct. Cortical nephron. So if you think about it, let's actually go back to this picture. With a cortical nephron, remember that cortical nephron, its entire tubular structure may be here within the cortex. So in this case, with that 300 osmolarity, are we going to be able to dramatically change the composition, the osmolarity of our blood, of our filtrate with these cortical nephrons?
No.
No, because if the surrounding osmolarity is only 300, again, the pumps will still pump stuff out. So we can still get rid of solutes, but we're not going to be able to majorly draw out water. So we can still get rid of solutes, but it's really this concentration gradient that is what allows us to reabsorb a lot of the water. We want to bring back those solutes, but we also want to bring back some of that water as well. And so this allows us to decouple those. So those 15%, and I know that doesn't sound like a lot, but remember when we say that you have 1 million nephrons in a kidney, 150,000 of these is a significant amount. So that, and that's really the other key to this too, and I'll go ahead and write it here. Remember, our goal at the end of this is to produce 1 to 2 liters of dilute urine. And this will allow us to do that. However, we only want to produce 1 to 2 liters of dilute urine if we are well hydrated. If we are not well hydrated, what do we want to do at that point?
Retain water.
Right, we want to reabsorb more water. And if we reabsorb more water, that is going to give us a very small volume of concentrated urine. So notice this loop of Henle allowed us to produce a small amount of dilute urine, but you've hit it on the head. If we want to produce concentrated urine, we need another mechanism. So these loops of Henle allow us to produce a small volume of dilute urine. But you're right, if we're not hydrated, there are times where we want to do more. We need to do more. And that more occurs in the distal convoluted tubule and the loop and the collecting ducts. Notice here is the process we just finished talking about it in this really nice, very simple illustration. We have our filtrate at about 300 milliosmoles. It's processed in the proximal convoluted tubule. Then, as we just saw, we are going to draw out a bunch of water, concentrating it very much. Then we're going to actively pump out a bunch of solutes, diluting it to about 100 milliosmoles. And the rest of the nephron, the collecting duct, is all impermeable. So our distal convoluted tubule and the collecting duct are impermeable to water. What that means is any water that is in the lumen when it reaches the distal convoluted tubule is going to stay in the lumen. And so as it stays in the lumen, it is going to produce our, and I know they say large volume, but again, we are going from 200 liters to 1 to 2 liters. So their large volume, we're talking about 1 to 2 liters of a very dilute urine. but you're not well hydrated. And when you're not well hydrated, as we mentioned, one of the things that happens is we produce anti-diuretic hormone. What anti-diuretic hormone does is it causes the expression of aquaporins in the distal convoluted tubule and those principal cells of the collecting duct. And when they are expressing those aquaporins, now they can reabsorb water. So let's think about this. Here in my end of my loop of Henle, I have diluted my filtrate to 100 milliosmoles. But then I open up a bunch of aquaporins in my distal convoluted tubule. What is the osmolarity of the cortex?
$300.
300.
300. So I've got 300 milliosmoles in my cortex and 100 in my filtrate. What's going to happen with those aquaporins now?
The water will go out till it reaches equilibrium.
Exactly. So notice by the time it reaches the collecting duct, we can get it to 300 milliosmoles. We can basically make it 1/3 of the volume it was before. But then this filtrate has to travel down the collecting duct. And where does the collecting duct go?
To the medullary papillae.
Yeah, it goes, which is at the end of the pyramid. So notice it travels through the pyramid. So as it travels through the pyramid, what happens to the osmolarity in our pyramid?
It goes up.
Yeah. As much as 1200, right? So notice as our filtrate continues down that collecting duct, more and more and more water can be pulled out to try to equalize to its environment till we get an incredibly small volume of water that has the same amount of solutes that it did before. And so now it has become concentrated. So notice this is twofold things that I want to emphasize. This concentration of the urine actually, I lied, 3 things. Occurs in the distal convoluted tubule and the collecting duct. Right. And in fact, you could argue more in the collecting duct than in the distal convoluted tubule because it passes through the medulla. And this is driven by hormones, primarily anti-diuretic hormone, but we know there are other hormones that can affect the reabsorption of water in our kidneys. And so unlike before, where we had our obligatory water reabsorption, where water had to follow the solutes, does the water have to come out here?
No.
No. In fact, water can only come out if it gets help. Water can only come out if it is facilitated. if it is given help by the expression of those aquaporins. So this is what we call facultative water reabsorption. So notice we have two different types of water reabsorption: obligatory and facultative, where it has to or where it only can with help. All right. Notice we can put all the pretty pictures together. Here again, we see that countercurrent mechanism, both countercurrent multiplier in the loop of Henle, setting up the concentration gradient and pulling the water out, and then that countercurrent exchanger from the Vasorecta. Those two countercurrent mechanisms that help us to produce our dilute urine. And then, as we talked about, we can then take that normal dilute urine, which stays dilute for the rest of the path through the distal convoluted tubule and collecting duct, or with the expression of aquaporins, thanks to anti-diuretic hormone, we can concentrate that urine with that facultative water reabsorption. Driven by hormones, can only be done with help, and occurs in the distal and convoluted tubule and the collecting duct. Whereas obligatory water reabsorption, remember, occurred in the proximal convoluted tubule. We've kind of already hinted at that. We know now that when we get to the distal convoluted tubule, we have absorbed most of the solutes. Again, 25% of the water has been absorbed, and we're now down to 10% of the original solutes. And the rest, as we mentioned, is hormone driven. Remember, as we said, in the distal convoluted tubule and the collecting duct, this is where we do the fine tuning. And our fine tuning is driven by hormones. Our hormones tell the kidney what we need, what we want to get rid of, what we want to keep. And this is where those changes take place. We've talked about lots of examples. Aldosterone led to sodium reabsorption. Where was that aldosterone produced again?
In the adrenal cortex?
Yep. Which of the three zonas of the adrenal cortex?
Glomerulus.
Yep, glomerulosa, absolutely. Right. Remember, it's called glomerulosa because it had ball-shaped structures, but remember, it also affects the kidney. So that kind of helps you to remember that as well, because we have a glomerulus here. So we talked about sodium, it drags a lot of stuff with it, including chloride. And then, like we said, this also helps us to get rid of excess potassium. This is not a pathway you need to worry about. So, but again, it just shows how it's very important to bring sodium and chloride back, but potassium has a much more ambivalent movement because, again, we want to keep low levels of it in the interstitial fluid and in the blood plasma. Parathyroid hormone tells the distal convoluted tubule to bring back what?
Calcium.
Calcium, excellent. Anti-diuretic hormone, we just did this, but tell me again, what does it tell the distal convoluted tubule to bring back?
Water.
Yeah, so notice we've talked about lots of hormones that target the kidney to tell us to bring something back, or conversely, to get rid of something. And here in the distal convoluted tubule is where we do that. So again, we're not going to worry about the specific pathways, but like I said, this is going to, pH is another important thing that is regulated hormonally in the distal convoluted tubule. And this is also where many of our wastes or toxins or antibodies or hormones or things like that will be released as well. And again, I'm not going to hold you responsible for the pathways. I think your book describes some of them, but I'm not going to worry about that too much. This is just where that fine tuning occurs. Again, remember to still filtrate in our collecting duct because it can be processed in our collecting duct. As we talked about, our principal cells are the ones where we get that facultative water reabsorption that occurs. It also, as you can see, plays a role in the absorption of sodium and the secretion of potassium. But remember, there was a second type, those intercalated discs, intercalated cells. Intercalated discs are found in the heart. Intercalated cells play a major role in our pH balance. And notice we keep kicking potassium out, but there are times when we don't have enough potassium and need to bring some back. And so they can help in that reabsorption if we get rid of too much potassium. And then lastly, finally, remember when it leaves the collecting duct, as was mentioned, out the papillae of the pyramids, oops, then it is finally urine. It's no longer processed. At this point, there's nothing in it that we want to bring back anymore. And we couldn't even if we wanted to. And here's your book's got a great in focus, great pretty pictures that describe this whole process. And again, put it all together into a nice, pretty bow. All right, questions on that? All right, we don't have too much more to go, but this is a good stopping point for our next break. Let's go ahead and take this as a nice transition point. We will go ahead and I'll leave you with my convoluted picture. Let's go ahead and take our last break, take a 15-minute break, come back at 2:27, and at 2:27, we will pick up from there. I'll see you guys in 15 minutes. All righty. Home stretch. As I mentioned, after all of this work, all of this processing that takes place from the moment it is first released from the glomerulus till the moment that it exits the collecting duct, we have processed 200 liters of filtrate to make 1 to 2 liters of urine. As we mentioned, it is mostly water, about 93 to 97%, and primarily nitrogenous wastes. Right? And then excess ions that we don't need. As we've talked about before, it has a yellowish coloration, typically a light straw color when it is dilute, caused by urobilin. If you remember, urobilin comes from bilirubin. Bilirubin, remember, we use from the breakdown of red blood cells to make our bile. And then remember, during that digestion of fats, that bilirubin is broken into stercobilin, which stayed in the digestive system, giving the brownish coloration to our feces, and urobilin, which was actually reabsorbed and released here in the kidneys. Typically, it is clear there should not be any kind of particulates in it. That can be an indication of a bacterial infection or things along those lines. It is slightly more dense than water. So if you, for instance, urinate in the middle of the night, or avoid urine in the middle of the night, I should say, and then don't want to make a lot of noise by flushing. So you go back to sleep, wake up the next morning and come in. For the most part, typically the urine will pool down at the bottom of your bowl. It will actually separate from the water, being slightly heavier than water. It is actually sterile when first produced. Now you have to be careful about this in two ways. I know when you walk around downtown Sacramento, where there's plenty of urine, sterile is not the thought that comes to mind when you take a sniff. But remember, we've got a very warm, very metabolically dense and also wet material here, which is a perfect breeding ground for bacteria. So much of that stench that we associate with voided urine is the bacteria from that. Also, again, it has to pass through the urethra. So often, for instance, as we've talked about before, when a doctor's trying to see what's going on with you and they take a urine sample, they don't want the first beginning of the stream as it comes out. You typically need to wipe the external orifice of the urethra, start the flow to cleanse the urethra, and then collect your sample midstream to be able to do that, because while the urine is sterile, your urethra is not. And typically it is acidic at about a pH of 6. But again, a lot of that has to do with what's going on in your body. And interestingly, one of the major factors that can influence the pH of your urine is your diet. If you're on that carnivore keto diet, it can become quite acidic as 4.5. And in fact, if you are vegan, in some instances, some vegan urine can actually become basic, getting closer to a pH of 8. All right. Questions on that? And again, typically it is slightly aromatic, but again, things like smell, things like color, are things that can be affected not just by our body, but also by medicines that we take, foods that we ingest. Can anybody give me an example of a food or a medicine that can change the color or the smell of your urine?
Asparagus.
There you go. That's the big one I was looking for. Absolutely. Asparagus absolutely tends to give a much more pungent aroma to your urine. And there are other things. There's some medicines that can make your urine more orangish in color, and so on and so forth. As well as medical conditions. We talked about jaundice. We talked about diabetes will make it smell much more sweet because of the glucose that's in there, things along those lines as well. All right. As I mentioned, 24/7, right? 7 days a week, all day long, you are constantly urinating, because urination is the formation of urine. However, urine, hopefully not 24/7, isn't leaking out of your body. And that is because of these structures here, your urinary tract. The goal of the urinary tract is one goal: store the urine till we get a socially appropriate location and then void it. I guess that's two functions, but those are the two functions of it: to store it till you get to a socially appropriate location and then void it. They do not process or modify the urine in any way. Once it leaves that collecting duct out the papillae of the medulla, it is unchanged for the rest of the time. The urinary tract contains 4 structures. The 2 ureters, those, remember, are retrograde organs that connect the kidney to the bladder. Notice these ureters actually come from the posterior side and actually move down and enter inferiorly. So if we were to draw this from the side, here is the bladder. The ureters don't connect to the top of the bladder. What happens is the ureter actually comes around posteriorly and inserts into the bladder at a posterior and inferior position. Why might that be beneficial?
Does that aid in kinking?
Absolutely.
Kinking of the ureters?
that definitely does help in two ways for the kinking, both in a good and in a bad way. As we increase the volume and the pressure increases, we know that our bladder is going to expand. As it expands, if the ureter connected to the top, as it pushed up, there's the chance that it could cause the ureter to kink, and that would absolutely be a bad thing. But as we also know, fluids don't like to be in a high pressure environment. So as the pressure builds up in the bladder, it may want to travel back up the ureter towards the kidney. And do we want that high pressure urine coming back into the kidney?
Oh,
No. So notice as this fills with urine, it is going to push down weight-wise, gravity-wise on that opening of the ureter and help to keep it closed. So it acts basically as a one-way valve. Now, doesn't that mean that the urine can't get into the bladder if it's being held closed by the weight? Well, how does urine get from the kidneys to the bladder? Gravity?
Peristalsis?
Peristalsis, exactly, right? Yeah, sure, we're standing or sitting upright most of the time, but that 8 hours of sleep you got last night as you're laying vertical, do you still want urine to be able to get to your bladder? If you're an astronaut in outer space with no gravity, do you still want urine to be able to get to your bladder? Absolutely, peristalsis. So because peristalsis are these powerful waves, when the peristaltic waves reach the end, they can squeeze the urine into the bladder, and then it collapses back down again. Excellent. So as I've said many times, pressure makes the world go round, and this is exactly an example of why this anatomy is convenient for that. So we have our two ureters, we have our singular bladder, and we have our singular urethra. We just talked about the ureters. Here we see them in their entirety. As I mentioned, they're retroperitoneal. As we just mentioned, they move urine via peristalsis. And we even talked about why they go to the posterior and inferior side of the bladder. What we didn't talk about is the histology. So let's take a look at the histology. This is, of course, a mucous membrane because it is open to the outside world. And in particular, the urinary tract. What is the epithelial tissue that lines almost the entire urinary tract?
transitional?
Exactly, that transitional epithelial tissue. Starting at the minor calyx, all the way through the major calyces, through the renal pelvis, through the ureter, through the bladder, and all the way through the proximal, 80% of the urethra. It is transitional epithelial tissue. The only difference is at the very distal end, where it is actually exposed to the outside world. What epithelial tissue do we have there?
Simple squamous?
If it's exposed to the outside world, would we want just a simple squamous there?
Non-keratinized stratified squamous.
There you go. You're right, we want flat cells, but we want many of them. A non-keratinized stratified squamous epithelial tissue. Very similar to what we saw in the digestive system, right? Pretty much it was simple columnar all the way to the very end, where it had a non-keratinized stratified squamous. And it's similar here as well. In fact, you're going to see a lot of similarities between the digestive system and the urinary system as we continue to move forward here. Here in the histology slide, we see a nice example of this transitional epithelial tissue, many cell layers. And as it moves towards the apical surface, the cells typically get bigger. So we have these big block cells on the apical surface. Again, we want that nice, big, distinct barrier. And I know this is a little magnification. I wouldn't use this on the exam, but remember when you're looking at this in a high magnification, a dead giveaway that you're looking at a transitional epithelial tissue is one of those apical block cells on the surface may have 2 nuclei. So if you see that, that's a dead giveaway. Notice again, all membranes have an epithelial tissue that sits on top of a connective tissue. Here is our connective tissue. And again, being a mucous membrane, this of course is an areolar connective tissue. And what would we call this areolar connective tissue based on its location?
Lamina propria.
Lamina propria. Here we see a thin layer, excuse me, of smooth muscle. And as we mentioned, because this is retroperitoneal, it basically has a fibrous connective tissue adventitia, which anchors it to the posterior wall of the abdominal pelvic cavity. Now, the one problem we have is that this is kind of the anatomy of all of the urinary tract. A mucous membrane with a transitional epithelial tissue, some smooth muscle, right? And so it's going to be kind of tricky sometimes to tell, especially the ureters and the urethra apart. The first thing that always helps me is how many ureters do you have?
2.
2. And notice it has 2 E's in it. How many urethras do you have?
1.
And notice it has 1 E in it. So that's spelling wise why I'm able to tell them apart. The second thing is obviously ureters are much, much smaller. Being much, much smaller, it's usually pretty easy to get an entire ureter on a slide so that we're going to be able to see it intact this way. What about a bladder? Are we going to ever see a bladder fully on a single slide for a microscope?
No.
No, even if you had a slide big enough to get it on a microscope, it'd be way too big to view the thing in its entirety on the microscope. So size is a big way to tell them. And then also when we get to the urethra is going to have a larger lumen. It has a larger lamina propria. I'm just going to abbreviate it LP there, with more glands, mucus glands, because it's closer to the outside world. And also have more elastic fibers in it as well. So those are things that we'll look for when we get to the urethra to tell the two of them apart. So this has a relatively small smooth muscle layer, relatively small lumen to it, and that is our ureter. Bladder, of course, by far is the largest of the organs. In some ways, it is similar to the stomach in that it is a large collapsible sac. It has folds in the mucous membrane known as rugae that again are going to allow for expansion just like the stomach. And also, just like the stomach, it has 3 distinct, okay, let me rephrase that. It has 3 layers of smooth muscle. However, in our stomach, those 3 layers were very, very distinct. We could clearly see the circular and the oblique, the circular and the longitudinal as we looked at it. In our bladder, the 3 layers are very intermixed with each other. So we are going to see a big, huge block of smooth muscle, but we're not going to be able to really tell the difference of the 3 layers. As such, the whole muscle collectively is just simply referred to as the Detrucer muscle. What's the job of this Detrucer muscle?
Contract.
Yeah, contract to void the urine using, again, peristalsis. Now, I know when you first start voiding the urine, that may not be as obvious, because the high pressure of the bladder, the high pressure of the urine inside the bladder, it tends to come in a fairly continuous stream. But as the bladder gets close to being empty near the end of your voiding, does it stay a continuous stream?
No.
No, it becomes more pulsatile in nature. And that more pulsatile in nature is because of those peristaltic contractions of the detrusor muscle. There are 3 openings to the bladder. The first 2 are the internal osses of the urethra, ureters, this is the urethral openings, and the internal os of the urethra. Notice in between these 3 regions is a, well, I don't like that color, is a thickened portion of the mucous membrane forming a funnel-like structure called the trigon. This rigidness serves 2, well, there's 2 reasons why we bring it up. First, it helps to keep this from collapsing, helps to keep the openings open so that urine can continue to come into the bladder and also can be voided from the bladder. From a clinical standpoint, this is significant because when you void urine from your body, do you think you are able to completely empty your bladder?
No.
No. So there can be some urine that, well, that can be, there is some urine that remains behind. As we mentioned, it is wet, it is warm, it contains metabolic resources. So most urinary bladder infections, when they occur, they typically start here in this trigon region. Because of that lack of voiding, again, it is the common location where urinary bladder infections will occur. Similar to what we saw in the digestive system, in that detrusor muscle, there is a circular enlargement that I'm completely exaggerating here of the circular muscle layer that goes around that internal os of the urethra. That is our internal urethral sphincter. It is, as we just mentioned, an extension of the smooth muscle. And as smooth muscle, does it have voluntary or involuntary control?
Involuntary.
Excellent. And just like we saw in the anus, we had an internal anal sphincter that was smooth muscle. What else did we have?
We had a secondary muscle that we could also control,
Yeah.
levator ani.
An external anal sphincter that was skeletal muscle and was an enlargement of the levator ani muscle. Well, here we have the exact same thing. We have an enlargement of the levator ani muscle, our urogenital diaphragm. Oops, no, go back. Down here. Ugh, why can I not grab onto that? Go back there. Huh, all right. Doesn't like it. All right, I'm gonna have to live with it be offset like that. We have a enlargement of skeletal muscle, an enlargement of the levator ani muscle that forms the external anal sphincter, external urethral sphincter, and it provides voluntary control. Again, just like we saw in the rectum and the anus. The only difference is, notice in the rectum, the smooth muscle and the skeletal muscle basically surrounded each other. They were in the same location. Notice here they're not. The internal is at the os where the bladder is, and the external is actually closer to the outer surface of the body. So they don't line up in the same way. Here we see the histology of the bladder. Remember, the bladder is intraperitoneal. As such, it has a serous membrane on its outer surface, and lo and behold, here we see that visceral peritoneum. Here we see our mucosa with, again, that transitional. And again, it doesn't, you can't tell at this magnification it's transitional. You just know that it is. And here we see that big, huge, thick, smooth muscle layer. Notice we can clearly see different orientations to different bands of muscle, but they don't form distinct layers the same way. They're very interwoven with each other. So we just collectively call these 3 layers of smooth muscle the Detrucer muscle. Notice it's also showing us some of the rugae, the big folds in the mucosa that we can see as well. All righty, this leads us to our urethra, which again is a single muscular tube that carries the urine out of the body. Again, via peristalsis, just like the bladder contracts with peristaltic waves, so does the urethra. And this voiding of the urine is not urination. Urination is the formation of urine. The appropriate anatomical term for voiding of urine is micturition. Right? Mastication, micturition, defecation, right? Fun with vocabulary. Notice, as I mentioned, its histology is similar to the ureters, but notice it has a much larger lumen. Notice to get the entire lumen on the screen in our microscope here, we have to be at a much smaller magnification where we're not actually able to see the individual cells of the transitional epithelial tissue. Notice being larger, it has more bigger invaginations of that lumen than the ureter did. That's another good indicator. As I also mentioned, it has a much, much thicker lamina propria, where we can see more glands, many more glands inside of this, where it's producing mucus, because again, we need that protection from the outside world. Here, the mucus isn't helping to smooth the transition of the urine the same way we saw with defecation. Now, this is here about capturing dust and debris and stopping that bacteria from getting in. Lastly, a much, much thicker smooth muscle wall as well. In fact, you can see 2 distinct layers to it, whereas you don't see that in the ureter. It won't always be this clear, but it's always going to be a much thicker smooth muscle layer. So of all of this, those are the 2 things that are probably the trickiest to tell apart, the ureter and the urethra. But if you pay attention to the size, if you pay attention to the size of the lumen, size of the lamina propria, size of the smooth muscle, that's really your big indication. All right. The last thing I want to talk about the urethra is that the urethra is one of the locations where we finally see some significant differences between male and female functions. Right? Again, when we were talking about the cardiovascular system, we talked about differences in sizes or amount of blood, differences in heart rates, respiration rates, all these kind of things that we've talked about that are differences for males and females and may mathematically be statistically significant, but aren't clinically significant. There's no real difference in function. Here is one of the major areas, and obviously when we get to the reproductive system next, we're going to see major differences between the male and female versions of things. So let's talk about those differences. Clearly, who has the longer urethra?
Males.
Males, absolutely. Right. In females, the length of the urethra is only about 1 inch. This is one of the reasons why in females, especially younger females are much more likely to get bladder infections than males are, because it's much easier, or the bacteria has a shorter distance they have to travel to get to the urine in that trigon region. Whereas the average length of the urethra, notice I said urethra length, not penis length, is 8 inches, about 20 centimeters. Whoops, drop my fidget. The locations are significantly different between males and females. As I mentioned, in males, it passes through the penis and also through one of the reproductive glands, the prostate gland. Females don't have a prostate per se, but it also doesn't connect to the reproductive system. The urethra is anterior to the vagina as opposed to passing through the vagina. So in the peritoneal region, or the urogenital diaphragm, pardon, not urogenital diaphragm, the urogenital triangle, in that region, there are 2 openings for females, one for the vagina, one for the urethra, whereas for the male, there's just 1 opening, and the penis for both. And that, of course, affects their function. For females, the urethra is solely a passageway in the urinary system. It only contains urine. Whereas males, the urethra is a pathway for both urine in the urinary system, but also for semen in the reproductive system. Your book's got some great pictures. Here are those examples. Here we see the females. Again, the anatomy here is a little bit basic, which is perfectly fine because it's going to be very complicated in the reproductive system. We have that short passageway from the bladder to the outside surface of the body. Again, remember here at the internal os is where our internal urethral sphincter is located. Here is that levator ani muscle helping to form our urogenital diaphragm. And here, that enlargement of it is our external urethral sphincter. So they don't line up the same way that it does in the digestive system. But other than that, the function is essentially identical. And then our external urethral orifice. Very different from the male. Here in the male, the urethra is so long and passes through so many structures that it actually is divided into 3 distinct regions. We can start with the bladder, and again, the enlargement of the smooth muscle, the internal urethral sphincter. And then there are 3 regions to the urethra in a male. There is the portion of the urethra that passes through this donut-shaped prostate gland, known as the prostatic urethra. There is the portion of the urethra that passes through the penis, known as the penile or the spongy urethra. And notice in between them is the very small portion that passes through the urogenital diaphragm. This happens to be where our enlargement of the levator ani muscle forming the external urethral sphincter passes. And so that tiny little bit of urethra that passes through the levator ani muscle passes through the external urethral sphincter is known as the membranous urethra. So for our male, we have 3 different regions, and another pretty picture that shows this as well. Here we see it in a sagittal section. And again, we have our prostatic urethra that goes through the prostate. We have our membranous urethra that passes through the levator ani muscle and through the external urethral sphincter. And then we have the penile or spongy urethra that passes through the penis.
Professor,
Yes.
is the spongy urethra also called the cavernous?
Yeah, cavernous cavernous urethra, penile urethra, spongy urethra, all are appropriate terms for it.
Got it. Thank you.
Yep. So again, why they felt the need to give a third name to this part that passes right through here, you got me for that, but that is there. But you can clearly see why they didn't bother naming any of the regions of the female, because basically it's an inch-long single structure that doesn't really pass through any other important structures, whereas this passes through 3 important structures, and so it gets 3 names: prosthetic through the prostate, membranous through the levator ani muscle or the urogenital diaphragm. And again, anatomist hatus. Prostate goes through the prostate. Penile goes through the penis, right? Levator ani, external sphincter. No, it's membranous for whatever reason. So there you go. Questions on that? Oh, and since we saw the sagittal view of the male, let's look at the sagittal view of the female. And this does a nice job of emphasizing the other component that we talked about, whereas the female's reproductive tract, the vaginal canal through the uterus, and then obviously through the uterine tubes to the ovaries, the reproductive tract is completely separate from the urinary tract, the urethra that leads to the bladder. And neither the two shall meet. 2 separate openings, whereas in the male, just 1. All right. Questions on that? All right, excellent. We have one thing left, our Micturition reflex. And our Micturition reflex basically is identical to the defecation reflex. So the defecation reflex you learned for the last exam is pretty much the exact same process you get to use for this one. We have a bladder. That bladder is connected to a urethra. And we have 2 sets of valves. Here in the walls of the bladder. Is our internal urethral sphincter, which as we know is smooth muscle, and so involuntary control. And whether you're a boy or a girl, you have the levator ani muscle. And in that levator ani muscle, you have an enlargement around the urethra. That is the external urethral sphincter. Which is made of skeletal muscle and provides voluntary control. Excellent. Like most homeostatic processes, this starts with a stimulus. What do you think the stimulus of this process is going to be?
Stretching of the bladder.
Exactly. Bladder fills to a specific point, and as it fills that specific point, it stretches and sends a signal. So we get urine filling this to a point. As it fills it to a point, it provides stretch. And this stretch produces a signal that is sent where? Well, let's think about this. Is it going to be a short reflex or a long reflex, do you think?
Long reflex.
Long reflex? Anyone think otherwise? I'm sitting waiting for an answer. So what do you think the correct answer is?
Short reflex.
Short reflex? Or if we think back to defecation, what did we have? Do we have a short reflex or a long reflex?
Didn't we have both?
Exactly. And we are going to have both here as well. The short reflex in this case is going to be to the bladder to start to contract. And this short reflex is going to relax the internal urethral sphincter. Notice when this relaxes, a little bit of urine can enter into the urethra. And when that little bit of urine enters into the urethra, are you aware of that?
Yes.
Yeah, absolutely. Between the contractions of the bladder and the urine entering the urethra, you become aware of the need to void. And this is when you start doing the pee-pee dance. Of course, when you feel that need to void, is urine flowing out of your body at that period of time? No, because that stretch of the bladder also stimulates a long reflex. That long reflex, again, goes through the pelvic splanchnic nerves to the sacral spinal cord. And this triggers the contraction of the external urethral sphincter. And so when this happens, we get the contraction of the external sphincter. So while we feel the need to void, while our bladder is contracting, urine doesn't come out of our body. But we're aware of the fact that we need to. So we go to a socially appropriate location. Or at least behind a bush. At which point, what do?
We voluntarily open the external urethral sphincter.
And now that both are open, voiding occurs. Now, just like defecation, there is a big key to this. Both sphincters have to be open for the voiding to occur, right? What if you were in the car feeling that need to void, but you just passed the sign that said the next rest stop is 30 miles? For the whole 30-mile drive, do you feel a constant need to urinate, to void, to micturate?
No, your body habituates.
Yeah. Right. As you're in time, your body will habituate to it. The contractions stop, the internal urethral sphincter will close, and the need to void goes away, which is a problem because when you get to the next rest stop, right, it can take you a moment or two to get things started again. However, there's a flip side to this as well. Remember, did I say it? I think I implied it, but this is, I'll just write it here in a different color to emphasize it. This is a parasympathetic reflex. So if instead of getting to a socially appropriate location, you went to a Oakland Raiders game. I know they're no longer in Oakland. They're at a big, super fancy stadium now in Vegas. But when they first moved back to Oakland and were at the Oakland Coliseum, before even the first round of renovations took place, did anybody go to a Raiders game there? At the old Oakland Stadium before it was, when they first moved back? I know you're all young, so probably not. And since most of you are females, you might not get this anyway. But for those of you who are males, you may be aware of this fact that basically the restrooms there, the urinals weren't urinals. It was basically a trough that everybody stood around and peed into. Now, for some individuals, standing in front of 25 other individuals, pulling out your penis and urinating at the same time that everybody else is, can make someone a little bashful or a little stressed. And if you have an increase of your sympathetic input from that stress, that of course suppresses your parasympathetic. And what happens?
You don't pee.
Yeah, that stress causes a closing of the internal urethral sphincter. And so even though you want to pee, you have trouble voiding the urine. Those people that are bashful, voiders, they are people who, again, that sympathetic stress causes problems with the relaxation of the internal urethral sphincter. So even though they're consciously relaxing the external, the stress keeps the internal closed and voiding can become a challenge.
Professor, wouldn't that be a sympathetic reflex?
The stopping, the bashfulness, the inability to, it interferes with that. So when you're stressed and you're having that, what's happening is that stress basically causes this. Remember, the relaxing of it was sort of the short reflex, but the parasympathetic and the sympathetic control smooth muscle. And so that input of the sympathetic causes this to close and stops the voiding. Yes, peeing when you're scared is a different type of mechanism. The cause of it is not fully appreciated, or the causality of it is not fully appreciated. It is believed that, and again, a lot of animals will do this. They will urinate or they will defecate in their flight response to try to confuse the predators with the smells and scents of that. So we know it is related to the mechanism of that. But yes, that is a different type of stress. In those cases, typically what's happening is you're getting an increase in pressure within the abdominal pelvic cavity, causing that voiding to occur from that. So that peeing when you're getting scared is a different type of mechanism. Yeah. All right. Now, again, habituation occurs, but as that habituation occurs, our bladder continues to fill. And as it continues to fill and push stronger, the contractions become more powerful. And eventually, can the contractions become powerful enough to force the urine out, whether you release the external urethral sphincter or not?
Yes.
Yeah, absolutely. So again, notice it's almost an identical process to what we saw in the, oops, I guess I have to delete this now. Almost the exact same process that we saw in our defecation. So that makes it easy. You learn to process once, you get to use it twice. And with that, we are done. That is everything you need to know for this section of the class. So, because we finished early, as always, and because we've covered a lot of material with these two organ systems, I am very, very happy to do a review. Let's take a quick 10-minute biology break, giving you a chance to micturate if you need to, or to grab some more water so that you don't concentrate your urine. And then we will come back in 10 minutes. So let's come back at 3:25, and at 3:25, I will happily answer any questions you guys have about the upcoming exam. So we will restart at 3:25. All right, see you guys in 10 minutes. All righty, let's go ahead and get started. Looks like right out of the bat, we have a great question about the oxygen hemoglobin disassociation curve. Well, let me answer this question first, and then I'll answer yours, Hannah. So, with the oxygen hemoglobin disassociation curve, we are measuring what? Let's define this first. Someone help me figure out what the point of this curve is.
A curve that compares percent saturation of hemoglobin to the partial pressure of the tissue, or excuse me, partial pressure of oxygen.
So it compares the amount of oxygen bound to hemoglobin and what?
And along with the partial pressure of oxygen.
Okay, excellent. Why? Why do we even care?
Because the lower the partial pressure of the tissue, the more active it is. So you will find less oxygen-bound hemoglobin in that area, meaning that you want more oxygen in that area.
Okay, excellent. So why this is important is absolutely it affects the amount of oxygen bound to hemoglobin in different environments. Excellent, right? This is what we want to know. And so again, this is all about transportation of our oxygen. So let's not even bother drawing with a curve, because again, you're not going to be able to draw a curve on the exam. So as you mentioned, it relates the percent of oxygen bound to hemoglobin. And what is our range for that?
You can only bind 4 oxygen per 1 hemoglobin.
Okay, so excellent. So perfect. So 1 hemoglobin can bind up to 4 oxygen. Now, of course, how much hemoglobin is in a single drop of blood?
Quite a lot.
Yeah, yeah, millions, hundreds of millions, absolutely. So when we talk about blood in general, we have a range, and that range is going to be from what to what? What is the range of oxygen bound to hemoglobin?
0 to 100.
Yeah. 0% to 100%. That makes sense. Excellent. The other thing is that it relates it to the partial pressure of oxygen in the blood plasma. All right. And what is the potential range, the biological range for that?
0 to 104.
Millimeters of mercury. Now, hopefully there's no place in your body where it gets down to 0, but biologically, technically that could happen, right? So at a very high level, then closer to the 100 to 104 range, how many oxygen bind to the hemoglobin?
4.
All right, and that would be 100%. And at a very low level, or no oxygen level, how many bind there?
0.
Yeah, which is our 0%. Now, normally when you have 2 points like that, you can draw a straight line. But there's one other factor that influences oxygen and hemoglobin. What is that one last factor?
Yeah.
Well, I was going to say that, like, I think you said the number one rule was that we can only bind as much oxygen as is available.
True. Yes. So again, that's what the curve shows us, right? So here, I know I said I wasn't going to draw it, but let's go ahead and draw it. If it is 0 oxygen available, then 0 is going to bind. If the partial pressure is 104, then basically we're going to get 100% binding, right? So we have 100% binding up here. We have 0% binding down here. We have 0 partial pressure here. And we have the partial pressure of oxygen equaling 104 here. And so when you have those 2 points, you can draw a line. Is that what our oxygen hemoglobin disassociation curve looks like?
No.
Well, it's in a name. It's a curve. Did I just draw a curve?
No.
No. So what curves it? What causes the curve?
When oxygen detaches or attaches to hemoglobin, it makes other oxygen molecules more likely to bind or remove from the hemoglobin, causing that curve.
There you go. Exactly. So when one oxygen binds, it causes the second oxygen to be easier for it to bind. So as a result of that, we get a curve that looks a little something more like that. So here is our oxygen hemoglobin disassociation curve. And basically, as you mentioned, as this points out, what this tells us is that how much oxygen binds to a hemoglobin is determined by how much oxygen is available. It's a pretty simple basic rule. All right. So then when we get back to this question of why it's important, the reason it's important comes down to this here. Let's say it this way. What determines how much oxygen is available in a particular environment?
The partial pressure.
Well,
that
the partial
environment?
pressure is how much is determined. I mean, how much is available. But what determines how much is available? That's how we measure how much is available. But what actually determines how much oxygen is available in an environment?
Yeah.
Metabolic activity.
Yeah, how much is used? In other words, like you said, the metabolic activity. So for instance, here, this is in the lungs. In the lungs, is there a lot of metabolic activity taking place?
No.
No. Basically, what's happening here is just gas exchange. So in this area of gas exchange, there's almost no metabolic activity that's taken on, because remember, it's a passive process. So it has a very high amount of oxygen. And when it has a high amount of oxygen, we can fill up the hemoglobin. However, the more active the tissue, the more oxygen, or the less oxygen, the more oxygen used, or the less oxygen available. So as our blood with that hemoglobin goes to areas with more and more use, less and less oxygen, what does our hemoglobin want to do?
Find more oxygen to it.
Does it want to hold on to more oxygen when it goes into those environments? Or if it's already filled up, what does it want to do to that oxygen?
Get rid of it.
Release them.
Yeah. Yeah. Hemoglobin goes to the bank and loads up on $20 bills. Right? And then as it's passing through, I don't know, wherever, someplace rich, Granite Bay. As it's passing through Granite Bay, does it need to be handing out too many $50 bills? Is it stopping Teslas on the side of the street and giving those poor people with their Teslas $50 bills?
No.
No, absolutely not. But as you continue to move further and further away, right through Roseville, in Roseville, they're a little bit poorer. They need a little bit more. Then you get out towards Rockland, and then from there you get out to Wheaton. And by the time you get whatever the heck's past Wheaton, Lincoln, and then Wheaton, right there, where it's all the farm people, then you're spreading $50 bills like it's going out of style. Right? The less oxygen that's available, the more oxygen you want to give that environment. And then you go back to the bank where there's plenty of oxygen and you pick up more. So basically, wherever the blood goes, it's going to give that tissue the correct amount of oxygen based on its level of metabolic activity. That is what this curve shows. Basically, the more oxygen an area is using, the more oxygen it needs. And so the less oxygen the hemoglobin will hold on to. All right.
Professor,
Yes.
you were also talking about like the different ways we can detect if
Don't ask that question yet.
sorry.
No, yeah. That's exactly where we're going, Leslie. I love that you were thinking ahead, but I don't want to confuse these two issues. So I want to make sure we do a hard stop right here and make sure we understand that, because you are correct. There is a whole nother level or an additional level we can add to this, but I want to make sure we understand this one before we go on to that one.
Gotcha.
But you're absolutely correct. That is exactly the absolute 100% appropriate question to ask next. So I love that you asked that. I just wanted to make sure we understand this first. All right, questions on this? All right, now, as Nathaniel very appropriately pointed out, as we talked about in class, whoops, hold on. Shoot, I didn't want to do that. Oh, no, all right, well, screw it. I'll use a different color. While how much oxygen is available is the primary way we tell how metabolically active a tissue is, there are other factors that can show how active a tissue is. We don't have to rely on just one factor, right? We're not going to just rely on where somebody lives. Just because someone lives in Granite Bay doesn't necessarily mean that they're rich, right? So we look at their car, or we look at their house, or we look at their clothes, right? There are other factors that can determine how active a tissue is. And I gave you 4 of them. What were the 4 other factors that could indicate how active a tissue was? Temperature, excellent.
Partial pressure of CO2.
Our waste product, pH. Now, again, the more carbon dioxide you have, the more acid you have, but pH is its own independent factor as well. And what's the 4th?
Hydrogen ion.
No, that would go along with pH. That one would directly relate to pH. But I like where you're thinking with that, because it is another example of a metabolic waste. So some other type of like metabolic waste, like nitrogen or biphosphoglycerate or something like that we talked about. All of these things can be things that indicate that even if the oxygen level isn't changing, that the tissue might be active. So let's think about this. If a tissue is more active, do we want to give it more or less oxygen?
More.
Want to give it more oxygen. So what that would mean is that on this curve, at every step along this curve, we would want to have less oxygen bound to the hemoglobin. We want to give more oxygen. We want to have less bound to the hemoglobin. And if we have less bound to the hemoglobin, the curve shifts down. Now, I emphasize that by just using 4 arrows. Where it gets a teeny bit confusing is if I connect all those dots from it curve coming down, it doesn't actually look like the curve went down, does it? What does it look like the curve does?
Shifted sideways.
Yeah, and more specifically to the right. So I will accept either of those 2 answers. You can say that the curve shifts down or the curve shifts right. They both say the same thing. They're saying that the tissue is more active and we want to give it more oxygen. We want to have less bound to the hemoglobin. Of course, that means the opposite is going to be true as well. If any of those 4 factors, like a decrease in temperature or a decrease in the partial pressure of CO2, or any of those other indicators tell us that the tissue is less active, then do we want to give that tissue more or less oxygen? We want to give it less oxygen. So we want to keep more bound to the hemoglobin. To do that, which way does the curve shift?
up or technically
Up,
like left?
exactly. So our curve is going to shift up. And you are also correct. When we then connect those dots, it looks like our curve has shifted to the left. So there you go. These are the ways that we can modify the oxygen hemoglobin disassociation curve to take in more information. So we can take in more information to make sure that the tissues are truly getting exactly how much oxygen they need. How much oxygen is available is going to be the primary determinant, but other things could be indicators that it needs more or it needs less. And so we want to take all those factors into consideration. So these 4 factors we just talked about, they can modify the curve. They're not needed for the curve. They modify the curve. All right.
So Professor,
Yes.
when we talk about the partial pressure of CO2 on this curve, if the partial pressure of CO2 is higher, then less binds or more?
So let's think about it. Let's talk about that case specifically. Partial pressure of CO2 increases. What do you think that indicates?
More
How do we-
activity.
Yeah, remember we make CO2 with cellular respiration. We use oxygen to completely break down our glucose, and as a waste product, we get CO2 and water. And of course, the reason we do that is to get energy to make ATP. So if we're making a lot of ATP to do a lot of work, we're going to get a lot more CO2. CO2, really, if you think about it, CO2 is just a fancy metabolic waste. It's just a gas metabolic waste, an important one, but it's still just a metabolic waste. So if there's more CO2, that tissue is more active. And if it's more active, then we want to give it more oxygen and it's going to shift down or to the right.
Got it. So we're talking about the partial pressure of CO2 of the tissue.
Yes, in the environment where the hemoglobin is.
Got it. Okay.
Yep.
Thank you.
Yep. Awesome. Excellent question. Any more questions on this one? Because I know it was a while ago now, but I know Hannah also at the same time asked a question as well. All right. So Hannah's question had to do with the urinary system. Okay. So what I would say is that to answer your question about, I'm not sure exactly what you mean by the renal vessels, but what you are definitely responsible for as a possible essay question, as we talked about, was the pathway for the secretion of hydrogen ions, which, remember, went hand in hand with the reabsorption of bicarbonate.
And sodium, right? Because then we need sodium as like an antiporter.
Yes, you do need sodium for that, but again, remember the point of this was to talk about the pathways. So yes, you are correct. This pathway starts with that antiporter using sodium to come in. But honestly, I wouldn't ask that on the question. Do you need to describe that? Absolutely. But the question you would see would be this one. Describe the pathway for the secretion of hydrogen ions and or the resorption of bicarbonate in the proximal convoluted tubule. right? And so you are right. If we were to describe that without the pictures, as you mentioned, that process starts with an antiporter on the apical surface of the simple cuboidal cell. And that antiporter on the apical surface of the simple cuboidal cell would who are you guys asking the questions to if it's not me? Who else is holding a review right now? On the apical surface, and again, so what this means that it uses the energy of sodium entering the cell to basically move hydrogen ions into the filtrate. Once it gets moved into the filtrate, what happens then?
It turns into carbonic acid
Excellent. Okay. So the hydrogen ion
with
binds.
bicarbonate.
Exactly. Binds with our bicarbonate to form carbonic acid. And from carbonic acid to water and carbon dioxide. What happens to that carbon dioxide?
Goes back in the membrane through simple diffusion.
Let's say it this way, crosses. Once it's in the cytoplasm, what happens to that carbon dioxide?
Reacts with water.
Carbonic acid again. Associates into hydrogen ions and bicarbonate. Hydrogen ions pumped out again. And what happens to the bicarbonate?
Uses a pump with sodium to get pumped out of the basal surface.
I always get mad at you guys for not being more specific, so let's be more specific here. So the hydrogen is transported out apical surface. Again, bicarbonate, along with sodium, is actively pumped out the basal surface, where it enters the blood. Well, enters interstitial fluid. And there you go. So yes, that could be something that could definitely be an essay question. All right. Now, does anybody have any questions for me instead of for some random other person who's in the chat? No. Great question. Someone was asking me about that in office hours today. So Exactly. If you, well, so let's start easy with this. Remember, these handouts were originally made for when we're in the classroom. And when we're actually in the classroom, we do actually have this really great slide in the classroom where a mouse kidney has actually been fully sectioned and is on a slide. So on a single microscope slide, we can see the entire kidney, which is pretty cool. Obviously, we can't do that with the human kidney under the microscope, but luckily for us, we have models of things like this, where we can see the cortex and we can see the pyramids and we can see the minor and the major calyces and the blood vessels and all that other anatomy that we talked about. So we can see those distinct regions of the kidney that you would see in a low-powered magnification view of this. Right, the same way that this next picture is kind of, if we increase the magnification of the microscope, we could see those components like the afferent and efferent arterioles, or the vasorecta, or the paratubular capillaries, or the proximal convoluted or distal convoluted tubule. Those are things that we could see here on a model. And in fact, our model even showed us a high magnification view of the renal corpuscle, where we could see the podocytes and their filtration slits, or we could see the glomerulus, or we could see the parietal layer of Bowman's capsule, or even the visceral layer of Bowman's capsule, the macula densa cells, the juxtaglomerular cells, right? The proximal convoluted tubule, all of those things that we could see that way. So these are all things that we've seen in models and we've seen in illustrations, and these are things that some of these are things that we could also see under a microscope as well. So if we were to look at the histology, identify the region of the organ in the field of view right here.
The renal corpuscle,
The region of the organ.
the cortex.
Mm-hmm. Exactly. Right. But you are right. The reason we know this is the cortex is because we have the renal corpuscle here. The renal corpuscle is only found in the cortex, so that's a dead giveaway. We are looking at the cortex here. And so again, we can see Bowman's capsule, we can see the glomerulus. We know this is the glomerulus and it's covered with podocytes, but do we happen to know whether that nucleus right there is the nucleus of one of the simple squamous endothelial cells of the glomerulus, or whether it's a podocyte?
No.
No, right? And nor am I going to ask it this way. As I've said before, I like this class because it's hard. And because it's hard, I don't have to be tricky. I can ask very simple, straightforward questions because I still know you're going to have to work really hard about it. And I will always use obvious examples of things. If I want to show you a podocyte, we've got that electron microscopy view or models or illustrations or other things that I could use for that. However, that doesn't mean you don't have to know any histology. And luckily, with some obvious examples of some stuff in here. For example, there is, what is that structure, do you think?
The afferent arteriole.
Not a bad guess.
Is that a proximal convoluted tubule?
Well, okay, I put the blue circle around that, and let's put a green circle around this. Of those two, which do you think is the proximal convoluted tubule and which one's the distal convoluted tubule? Green or blue? Which one's proximal?
Green,
How do you know?
because the proximal has the highest amount of microvilli, and so you can kind of see a blurriness in the middle.
Yeah, not only, remember, does it have more microvilli, but they're longer as well. So the lumens of these look very, very dirty. This one, or this one here, or this one over here, notice their lumens are very clear. And so the ones with a very clear lumen are the distal convoluted tubule, and the ones that are more dirty looking on the inside, fuzzy looking on the inside, are the proximal convoluted tubules. So yes, some of these things are things you are responsible for histologically as well. Right, and so obviously this would be a high magnification view, whereas, like I said, that we don't have a lower magnification view of it here histologically. We have one in the classroom. We don't have one here though, but the model gives us that whole view of the kidney. All right. Great question, Sean. Any others? All righty. Well, I will remind you, I also have office hours tomorrow from noon 30 to 1:30. I am always available by email. Otherwise, study hard, and I will see you guys on Thursday for your exam. Well, I won't see you, but take your exam on Thursday, and I will see you 1 week from today. Hopefully, I won't see you Thursday. The only reason I see you Thursday is you have a problem with the exams. So hopefully, I will not see you Thursday. Take care. Good luck, and I will see you guys next week.
Thank you.